What if life on Mars didn't start there at all? What if it arrived from somewhere else entirely, carried across the darkness of space by a visitor from beyond our solar system? In this story, we're stepping into an imagined version of our cosmic neighborhood, a what-if universe built on real physics, but not describing real events in our world. That sounds impossible, doesn't it? Life traveling between worlds, hitching a ride on an icy wanderer that's journeyed through interstellar space for millions of years. Yet in our solar system, this exact scenario may have already played out, and scientists are taking it seriously enough to study it right now. Something remarkable happened when 3I Atlas, an interstellar comet from beyond our solar neighborhood, made an unusually close pass by Mars. This wasn't just another space rock drifting through our cosmic backyard. This was a visitor from another star system, carrying material that formed in conditions utterly different from anything in our solar system, approaching the red planet at precisely the right distance and circumstances to make scientists wonder about something extraordinary. Think about what that means for a moment. A comet born in the space between stars, forged in the remnants of some distant stellar system, travelling through the cold darkness for eons, arrives at Mars at just the right time, just the right distance, releasing material as it heats up from our sun. Could that material have reached Mars? Could something within it have survived the journey? Could life itself travel this way? In this scenario, these aren't just science fiction questions. They're the kinds of mysteries that space scientists in this imagined future would be desperate to investigate. In this imagined universe, the encounter between 3I Atlas and Mars becomes a natural experiment, a perfect setup to explore whether the transfer of biological material between worlds could be more than theory and might actually occur. You might be wondering why this matters so much. After all, we've been searching for life on Mars for decades, sending rovers, landers, and orbiters to study the planet's surface and atmosphere. We've found tantalizing hints: organic molecules, methane fluctuations, ancient river valleys that once flowed with water. But the question of whether Mars ever harbored life, or still does in some hidden refuge beneath its surface, remains one of the greatest mysteries in science. What if we've been asking the wrong question? What if the question isn't just whether life arose on Mars independently, but whether life might have arrived there from somewhere else? What if Mars and Earth, and perhaps other worlds in our solar system and beyond, are connected by biological threads we're only beginning to recognize? This concept has a name in science: Panspermia. It's the hypothesis that life, or at least the building blocks of life, can travel between worlds, spreading throughout a solar system or even between star systems. For decades, it was considered an interesting idea but difficult to prove. Now, with the discovery of interstellar objects like 3I Atlas passing through our solar system, with our growing understanding of extremophile organisms that can survive incredible conditions, with robotic missions in this story designed to scoop up Martian rocks and soil and return them to scientists, we can imagine having the tools to truly test whether this kind of transfer ever occurs. The close encounter between 3I Atlas and Mars wasn't planned by anyone. It was a cosmic coincidence, a natural alignment that, in this story, gives scientists a realistic scenario to study. Unlike a simple thought experiment, this scenario imagines an interstellar object interacting directly with a planet in real time, under realistic conditions, with all the same physical processes that would operate in our universe. Scientists tracked 3I Atlas carefully as it approached the inner solar system, watching as solar radiation heated its icy surface, causing it to outgaze, to release jets of vapor and dust that create the characteristic tail we associate with comets. As it swung past Mars at a distance far closer than Earth's orbit, that outgassing was happening actively. Material was being released into space. The gravitational field of Mars was present. The thin Martian atmosphere was there, however tenuous. All the ingredients were in place for a potential transfer of material. Whether that transfer actually happened, whether any material from 3I Atlas entered Mars's atmosphere, whether anything within that material could have survived the radiation, the temperature extremes, the violent deceleration, these are questions that require evidence. And that's what makes this so compelling. We're not just spinning wild fantasy, we're following a carefully constructed scenario grounded in real physics, and imagining how future missions in this universe might one day test whether anything like it truly happened. I want to take you on a journey through this question. We're going to explore what we know about 3I Atlas, what happened during its encounter with Mars, what science tells us about the possibility of life travelling through space, and what it would mean for our understanding of life in the universe if comets really can deliver biological material between worlds. We'll look at real research, real missions, real data, and we'll be honest about what's established fact versus what's educated speculation. This isn't about sensationalism. It's about following the evidence wherever it leads, asking the questions that the evidence raises, and understanding that some of the most profound discoveries in science come from noticing unusual events and investigating them thoroughly. In this story, the passage of 3I Atlas near Mars is exactly that kind of event. Unusual, unexpected, and potentially revealing. If you're curious about how life might spread through the cosmos, if you've ever wondered whether we're truly alone or whether the universe is more connected than we imagine, I'd love to have you join this exploration. Hit the like button so this reaches others who share that curiosity, and drop a comment telling me where you're watching from, because this question reaches beyond any single place on Earth. It's about all of us, our origins, and the nature of life in a vast and mysterious universe. Let's begin by understanding what 3I Atlas actually is, because that's where this story truly starts. With a visitor from another star system entering our cosmic neighbourhood and changing the questions we ask about life itself. To understand what makes the encounter between 3I Atlas and Mars so scientifically intriguing, we first need to understand what 3I Atlas actually is, and why interstellar objects are fundamentally different from everything else in our solar system. When we look up at the night sky, almost everything we see, the planets, asteroids, comets, and moons, form together from the same disk of gas and dust that created our Sun. They're all siblings, born from the same cosmic cloud, sharing the same chemical composition, shaped by the same stellar environment. They orbit our Sun because they've been gravitationally bound to it since their formation. They're part of our solar family. Interstellar objects are not. They come from somewhere else entirely, formed around different stars under different conditions, with different chemical compositions reflecting the unique environments of their birth systems. They travel through the space between stars, unbound by any sun's gravity, wandering through the galaxy on hyperbolic trajectories that carry them through solar system after solar system. When one of these visitors passes through our neighbourhood, it's not just another comet, it's a messenger from another part of the galaxy, carrying information about distant stellar systems we may never reach. 3I Atlas is one of these visitors. The 3I designation tells you immediately what it is. The third confirmed interstellar object detected passing through our solar system. The first was Oumuamua, that cigar-shaped mystery that sparked intense debate in the scientific community. The second was Borisov, which looked and behaved more like a typical comet despite its interstellar origin. 3I Atlas became the third member of this exclusive group, spotted by a wide-field sky survey, a network of telescopes designed to catch wandering objects and potential hazards in this imagined solar system. But 3I Atlas had something the others didn't. Its trajectory brought it remarkably close to Mars, creating a natural experiment that astrobiologists could actually study. This wasn't a distant flyby observable only through telescopes. This was a close encounter where real physical interaction between the comet and a planet became possible. What astronomers discovered about 3I Atlas tells us a great deal about its nature and origin. Like all comets, it's primarily composed of ice, water ice, along with frozen carbon dioxide, methane, and ammonia. But interstellar comets can have different ratios of these materials, different trace elements, different organic compounds reflecting the chemistry of whatever stellar nebula birthed them. In this scenario, early spectroscopic scans of 3I Atlas appear to hint at complex organic molecules, the kind of carbon-based chemistry astronomers often expect to find in icy wanderers like this, the carbon-based compounds that form the foundation of life as we know it. That alone doesn't mean anything biological. Organic molecules form naturally in space all the time through purely chemical processes, but it establishes that the building blocks were present. The trajectory of 3I Atlas is what really sets it apart. Objects orbiting our Sun move on elliptical paths, bound by solar gravity, returning again and again in predictable cycles. Interstellar objects follow hyperbolic trajectories. They come in from outside the solar system, curve around the Sun as its gravity bends their path, then head back out into interstellar space, never to return. The mathematics of the trajectory tells astronomers definitively whether an object is bound to our Sun or just passing through 3I Atlas was clearly a visitor. travelling far too fast and on a path too open to ever be captured by our solar system's gravity. That speed matters enormously. Interstellar objects travel faster than native solar system bodies because they're not slowed by gravitational interactions with the Sun over billions of years. 3. I-Atlas was moving at velocity high enough that its journey through the inner solar system would be brief, measured in months rather than years. It plunged inward from a distant region of interstellar space, curved around the Sun, brushed past Mars during that brief passage, and then raced back out into the dark between the stars, leaving our neighbourhood as mysteriously as it arrived. What happened as 3? I-Atlas approached the Sun is where things get interesting for our question about Mars. Comets are often described as dirty snowballs, chunks of ice and dust that spend most of their existence frozen solid in the cold darkness of space. But when a comet approaches a star, solar radiation heats its surface, causing the ice to sublimate, to transform directly from solid to gas without melting. This outgassing creates the spectacular tails that make comets beautiful, and also releases dust, organic compounds, and whatever else is embedded in the ice. For 3I Atlas, this outgassing began as it crossed into the inner solar system and intensified as it approached closer to the Sun. By the time it reached the orbital distance of Mars, it was actively releasing material. Telescopic observations confirmed this, showing the characteristic coma, the cloud of gas and dust surrounding the comet's nucleus. Growing larger as solar heating increased, material was streaming off the comet in jets, pushed away by solar radiation pressure and the expansion of sublimating ice. This is crucial to understanding the potential for material transfer. A comet isn't just a solid object flying past a planet, it's actively shedding material, creating a trail of gas and dust that spreads out along its orbital path. As 3I Atlas approached Mars, it was leaving behind a dispersed cloud of cometary material through the region of space that Mars was also travelling through. The timing of this encounter matters as much as the distance. Mars and 3I Atlas weren't just nearby in space, their orbits intersected in time as well. Mars, moving in its regular elliptical orbit around the Sun, was at the right point in its orbit at the right time to potentially pass through the material trail left by the comet. This kind of alignment isn't common. Most objects passing through the solar system, even those that come relatively close to planets, don't intersect in both space and time in a way that allows for material exchange. Scientists who track near-Earth objects and potential asteroid impacts understand these principles well. It's not enough for two objects to have orbits that cross. They have to cross at the same time. The fact that 3I Atlas and Mars achieved this alignment made the encounter scientifically valuable, regardless of whether any material transfer actually occurred, because, in this imagined universe, it gives researchers a concrete case study to analyze with simulated and observational data rather than just abstract models. The chemical composition of 3I Atlas, based on spectroscopic analysis, showed signatures consistent with formation in a different stellar environment than our own. Certain isotopic ratios, certain molecular species, certain dust properties suggested that this comet formed in a region with different chemical abundances than the disk that created our solar system. This means that any material from 3I Atlas would carry a distinct chemical fingerprint, potentially identifiable if it ever ended up in Mars's atmosphere or on its surface. Think about what this represents. Here was an object carrying material from another star system, material that encoded information about that distant system's chemistry and conditions, approaching Mars while actively releasing that material into space. The physical setup was there. The question became, what does physics tell us about whether any of that material could actually reach Mars? What would it take for cometary material to be captured by a planet's gravity, to survive the intense radiation of space, to make it through an atmosphere without burning up completely? These aren't simple questions, and answering them requires understanding some fascinating physics and chemistry. Let's examine exactly what happened during that close approach to Mars, because the devil, as always, is in the details. The encounter between 3I Atlas and Mars was, in astronomical terms, remarkably close. When we talk about cosmic distances, close is relative. Space is so vast that even nearby objects can be separated by distances that dwarf anything we experience on Earth. But by the standards of planetary encounters with comets, this was genuinely close, close enough that scientists took notice and began calculating whether physical interaction was possible. Mars, orbiting the Sun at a distance much greater than Earth, but still within the inner solar system, has a thin atmosphere, less than 1% the density of Earth's, that might seem like it would make Mars less capable of capturing material from passing objects. And in some ways, that's true. But a thin atmosphere also means less resistance, less heating from friction, and potentially more opportunity for material to reach the surface intact if it's captured at all. The question wasn't whether Mars could catch everything. 3I Atlas released. It was whether it could catch anything. The physics of how this might work involves several interconnected processes. First, there's the question of relative velocity. When two objects in space pass each other, their relative velocity, how fast one is moving compared to the other, determines what kinds of interactions are possible. If the relative velocity is too high, material from one object simply races past the other too quickly to be captured. Gravitational attraction requires time to work, time for the planet's gravity to bend the trajectory of particles and pull them in. At extremely high velocities, particles zip through a planet's gravitational field before that field can significantly alter their paths. 3I Atlas was moving fast. All interstellar objects are. But the geometry of its trajectory and Mars's orbit meant that during the closest approach, the relative velocity was lower than it might have been under different circumstances. This created what physicists call a capture window, a range of conditions where gravitational capture becomes theoretically possible for at least some percentage of the material being released. As 3I Atlas heated up from solar radiation during its approach, the outgassing we discussed earlier was releasing material at various velocities. Some particles were ejected quickly from the nucleus by the force of sublimating ice, moving fast enough to escape the comet's own weak gravity. Others drifted away more slowly, carried by expanding gas. This created a distribution of velocities among the released particles, a spectrum ranging from slow-moving dust grains to fast-moving gas molecules. The slow-moving particles are the interesting ones for potential capture. A dust grain or small ice particle drifting away from the comet at relatively low velocity could, in principle, have its trajectory significantly altered by Mars's gravity as the planet passed through or near the comet's outgassing region. The gravitational force wouldn't be dramatic. Mars is smaller than Earth, with weaker gravity, but it doesn't need to be dramatic. It just needs to be enough to bend the particle's path toward the planet rather than past it. Now let's consider what happens if a particle's trajectory does bend toward Mars. As it approaches the planet, it accelerates, falling down Mars's gravity well and gaining speed. This is where the atmosphere becomes important. A particle approaching Mars from space enters an increasingly dense atmospheric environment, though dense is relative when we're talking about Mars's tenuous air. The upper atmosphere of Mars is extremely thin, consisting mainly of carbon dioxide at pressures that would be considered a hard vacuum by Earth standards. When a particle enters this upper atmosphere, several things start happening simultaneously. Friction with atmospheric molecules, however sparse, begins to slow the particle down and heat it up. The faster the particle is moving, the more intense this heating becomes. Meteors on Earth create brilliant streaks of light because they're travelling so fast that atmospheric compression heats the air in front of them to thousands of degrees, vaporizing the meteor itself and creating the flash. We see Mars's thin atmosphere produces less dramatic heating, but the principle is the same. This is where size becomes crucial. Large particles entering fast burn up quickly, vaporizing from the intense heat before reaching the surface. But smaller particles, especially those that enter at shallower angles or lower velocities, can survive atmospheric entry. Dust particles, ice grains containing organic molecules, tiny fragments of cometary material, these could potentially survive the passage through Mars's upper atmosphere and drift down to the surface, particularly if they're entering at velocities lower than typical meteors because they've been slowed by gravitational capture rather than falling straight in from interstellar space. The outgassing from 3I Atlas during its approach to Mars was continuously releasing material over time, not in one burst but as a steady stream as different parts of the comet's surface were heated by the sun. This meant that Mars, as it moved through its orbit, could have encountered this material stream for an extended period, potentially hours or even days, depending on the geometry of the encounter and the dispersion of the outgassing cloud. Scientists studying the encounter used orbital mechanics to calculate the probable distribution of cometary material in space during the time Mars was closest. These calculations involve complex physics, modelling how gas and dust disperse from a sublimating comet nucleus, how solar radiation pressure pushes particles outward, how the comet's own weak gravity and rotation affect material distribution, and how all of this evolves over time as the comet moves along its trajectory. The result is a three-dimensional map of where cometary material probably existed in space at various times, and whether Mars's orbital path would have taken it through any of these regions. In this imagined encounter, the best model runs suggest that Mars may have passed through regions where material from 3I Atlas was drifting, raising the possibility, but not the certainty, of some interaction. The question then becomes quantitative. How much material, how dense was it, what was its composition, and what percentage might have been captured? These quantitative questions are harder to answer precisely because they depend on details we can't measure perfectly from Earth. We can estimate the outgassing rate of the comet based on how bright it appeared, and how its brightness changed as it approached the Sun. But converting brightness to actual mass loss involves assumptions about the comet's composition and structure. We can model atmospheric capture on Mars, but the models have uncertainties depending on poorly known parameters, like the exact density profile of Mars's upper atmosphere at the time of encounter. What we can say, even in this fictional setup, is that the physics make such an exchange possible in principle. The encounter was close enough, the velocities were in a range where capture is theoretically possible, material was being released at the right time, and Mars was in the right place. Whether capture actually occurred, and in what quantities, is a question that may require direct evidence from Mars itself to answer definitively. There's another factor worth considering. Mars's thin atmosphere isn't uniformly distributed. It varies with altitude, of course, but also with latitude, time of day, and season. Mars has seasons because its rotation axis is tilted similar to Earth's. The planet's atmosphere can be slightly denser or thinner, depending on whether it's summer or winter in a given hemisphere, whether dust storms are occurring, and other factors. The time of year on Mars when 3I Atlas made its closest approach would have influenced how the atmosphere interacted with any incoming material. All of these physical factors — gravity, velocity, atmospheric density, particle size, entry angle — combine to determine what happens during an encounter like this. None of them individually is sufficient. They all have to align properly for material transfer to occur. The remarkable thing about the 3I Atlas Mars encounter is that they did align at least closely enough to make the possibility real rather than purely hypothetical. But there's a deeper question here that goes beyond the physics of this one encounter. Even if material from 3I Atlas did reach Mars, even if dust grains or ice particles made it through the atmosphere and settled onto the surface, why would that matter biologically? What is it about comets that makes scientists think they could carry anything living, or capable of becoming living, across the void of space? That question takes us into some of the most fascinating research in modern biology and astrobiology, the science of extremophiles and the hypothesis called Panspermia. The idea that life might travel between worlds isn't new, and it's not fringe science. It has a name, Panspermia, and it has a legitimate place in scientific discussion about the origins and distribution of life in the universe. What has changed over the decades is that we now have enough knowledge about extreme survival, enough evidence from space missions, and enough understanding of the early solar system to evaluate whether panspermia is merely possible or actually likely. Panspermia, in its simplest form, proposes that life, or at least the organic building blocks of life, can spread from one location in space to another. There are different versions of this hypothesis. Lithopanspermia specifically refers to life spreading via rocks, material ejected from one planet by asteroid impacts that travels through space and eventually lands on another world. Cometary panspermia suggests that comets, which contain ice and organic compounds, might serve as vessels carrying biological material between star systems or between worlds within a solar system. The hypothesis has serious scientific pedigree. Francis Crick, who helped discover the structure of DNA, considered panspermia seriously enough to write papers about it. Fred Hoyle, the renowned astronomer, proposed that comets might deliver organic compounds to Earth. More recently, scientists like Chandra Wickramasinghe have conducted research examining the possibility that microbial life could survive the conditions of space and atmospheric entry. These aren't fringe figures. They're respected scientists who recognize that panspermia deserves rigorous investigation, not dismissal. The reason panspermia deserves this serious consideration comes down to several converging lines of evidence. First, We now know that organic compounds are common in space. Meteorites that fall to Earth frequently contain amino acids, the building blocks of proteins. The Murchison meteorite, which fell in Australia, contained over 70 different amino acids, many of which don't occur naturally on Earth. These molecules formed in space, survived the journey through Earth's atmosphere, and arrived intact. If simple organic molecules can make this journey, what else might be able to? Second, we've discovered that life is far more resilient than anyone imagined decades ago. Extremophiles, organisms that thrive in conditions that would kill most life, have revolutionized our understanding of biology's limits. Scientists have found bacteria living in boiling hot springs, in the frozen ice of Antarctica, in acidic lakes that would dissolve human flesh, in the radioactive waste pools of nuclear facilities, and even miles underground where no sunlight ever reaches. Life adapts to conditions we once thought utterly incompatible with biological processes. Some extremophiles are particularly relevant to panspermia. Tardigrades, microscopic animals also called water bears, can survive being frozen, being heated to extreme temperatures, being subjected to radiation doses thousands of times higher than what would kill a human, and even exposure to the vacuum of space. Scientists have sent tardigrades into orbit, exposed them directly to space outside the protection of a spacecraft, and brought them back to find that many survived and even reproduced afterward. If a complex organism like a tardigrade can survive space, what about simpler, more resilient bacterial spores? Bacterial spores are another crucial piece of evidence. Certain bacteria, when stressed by environmental conditions, form spores, highly resistant dormant structures that can survive for extended periods under conditions that would kill active bacteria. Spores have been revived after thousands of years in ice, after heating, after radiation exposure. Experiments in low-orbiting laboratories and on uncrewed platforms in this universe have shown that bacterial spores can endure exposure to space for surprisingly long periods, radiation, extreme cold, and vacuum for extended periods, months to years in controlled experiments. The question then isn't whether life can survive space conditions. We know some organisms can, at least for a while. The question is whether life could survive the complete journey from one world to another, including ejection from the source planet, potentially millions of years in space, exposure to radiation during that journey, and the violent entry through an atmosphere and impact with a surface. This is where lithopanspermia research becomes relevant. When large asteroids impact a planet, they can eject material into space at velocities high enough to escape the planet's gravity. Calculations show that rocks have been exchanged between Earth and Mars throughout solar system history. We've found meteorites on Earth that came from Mars, ejected by impacts and eventually falling to Earth. If rocks can make that journey, could microbes inside those rocks survive? Laboratory experiments have subjected bacteria to impact shocks simulating asteroid strikes, subjecting them to pressures and accelerations thousands of times Earth's gravity. Some bacteria survived. Other experiments have embedded bacteria in rock samples and exposed them to simulated space conditions, radiation, vacuum, temperature extremes. Some survived those conditions as well, at least for the duration of the experiments. While no single experiment has proven that life could survive the complete journey from one planet to another, each piece of evidence suggests that the stages of such a journey are survivable by at least some organisms. There's also the question of radiation. Space is filled with high-energy particles, cosmic rays and radiation from the sun, that damage biological molecules. Particularly DNA-extended exposure to this radiation would kill most life. But there are protective mechanisms. Being embedded inside a rock provides shielding, ice provides shielding. Radiation damage decreases exponentially with depth into a material, so organisms even a short distance beneath the surface of a meteorite or cometary ice might receive much lower radiation doses than those on the surface. For bacteria in spore form, with minimal metabolic activity and highly condensed DNA, the radiation tolerance is higher than for active organisms. The timeline matters too. A journey from Mars to Earth, or Earth to Mars via rock ejected by impact, typically takes thousands to millions of years based on orbital mechanics. That's a long time to survive in space. But bacterial spores on Earth have been revived after thousands of years, and there are mechanisms by which organisms in deep dormancy could potentially remain viable for much longer periods. The cold of space actually helps in this regard. Chemical degradation slows dramatically at temperatures near absolute zero. What about interstellar travel, like 3i Atlas made? That's a harder question because the timescales extend to millions or potentially billions of years. No experiment can directly test whether organisms could survive that long in space. But the hypothesis isn't necessarily that living, active organisms survive the journey. It might be that organic molecules survive, complex compounds that could contribute to the emergence of life on an arrival planet even if they weren't alive during transit. Or it might be that extremely dormant spores, protected deep within ice, remain viable longer than we can currently test. One thing is certain, we can no longer dismiss panspermia as impossible. It's not proven, but it's not impossible. It's a hypothesis that has moved from the realm of speculation into the realm of testable science. We have the tools to examine returned samples from asteroids and comets for organic compounds and even preserved biological materials. We have experiments that can simulate longer and more realistic space journeys. We have missions to Mars that can search for biosignatures and analyze the chemistry of Martian rocks and atmosphere. The question isn't whether panspermia violates any known physical or biological laws. It doesn't. The question is whether it actually happens, and how frequently, and whether it has played a role in the distribution of life in our solar system or galaxy. Those are empirical questions, answerable through observation and experiment, and that's what makes the encounter between 3i Atlas and Mars so valuable. It gives us a specific, real-world case to examine, to see if the theoretical possibility translates into actual reality. So let's apply what we know about panspermia, about survival in space, about cometary composition and Mars's environment, and ask, could the 3i Atlas encounter actually have resulted in the transfer of biologically relevant material to Mars? What would the requirements be, and did they exist? Now we come to the heart of the question. We've established that 3i Atlas made a close approach to Mars, that comets release material through outgassing, that panspermia is theoretically possible, and that some organisms can survive harsh space conditions. But could this specific encounter have resulted in actual transfer of biologically viable material from the comet to Mars? Let's work through the requirements systematically. First, material has to actually leave the comet in a way that allows it to encounter Mars. Outgassing does this naturally. As ice sublimates, it releases dust particles, organic compounds, and everything else embedded in the ice. But the velocity at which material is released matters. If it's ejected too fast, it has too much energy to be gravitationally captured by Mars. If it's released too slowly, it might remain gravitationally bound to the comet itself and never separate enough to encounter Mars independently. Observations of cometary outgassing show that ejection velocities vary widely. Gas molecules move fastest, often at speeds of several hundred meters per second, as they escape the comet nucleus. Dust particles move slower, typically at speeds ranging from a few meters per second up to hundreds of meters per second, depending on particle size and the violence of the outgassing event. Smaller, lighter particles can be accelerated by the escaping gas and pushed to higher velocities. Larger, heavier particles tend to drift away more slowly. For Mars capture to be possible, we need particles released at velocities low enough that Mars's gravity can alter their trajectory significantly. Mars's escape velocity is around 5 kilometers per second. That's the speed you'd need to completely escape Mars's gravity well from the surface. At the distance where 3i Atlas passed by, the gravitational pull is weaker, but it's still sufficient to deflect the paths of particles moving at speeds much lower than escape velocity. A dust particle drifting away from 3i Atlas at, say, a few dozen meters per second could have its path bent toward Mars if the geometry is right. Solar radiation adds complexity here. Sunlight exerts pressure on small particles, a phenomenon called radiation pressure. For tiny dust grains, radiation pressure from the Sun can actually be stronger than the gravitational pull from a planet like Mars, pushing particles outward away from the Sun and potentially away from Mars. But radiation pressure decreases in effectiveness for larger particles, and it depends on composition. Dark, light-absorbing particles experience different forces than bright, reflective ones. The interplay between Mars's gravity and solar radiation pressure creates a size range of particles most likely to be captured. Not too small that radiation pressure dominates, not too large that they don't separate from the comet in significant numbers. Let's say some particles do get captured by Mars's gravity and begin falling toward the planet. Now they face atmospheric entry, and this is where things get challenging. As a particle accelerates toward Mars, it gains speed falling down the gravity well. By the time it reaches the upper atmosphere, it's moving at several kilometers per second. Not as fast as a typical meteor, which might be moving at tens of kilometers per second, but still fast enough to generate significant heating from atmospheric friction. The heating during atmospheric entry is what destroys most meteors. The bow shock in front of the meteor compresses atmospheric gas, and compressed gas heats up dramatically. This heat radiates back to the meteor, heating its surface to thousands of degrees. Most of the meteor vaporizes, creating the brilliant streak of light we see. Only the largest meteorites, or those coming in at shallow angles that extend the entry time and reduce peak heating, survive to reach the surface. Mars's thin atmosphere works both for and against survival. On one hand, lower atmospheric density means less friction and less heating for a given velocity. A particle entering Mars's atmosphere at the same speed as one entering Earth's would experience significantly less heating on Mars. On the other hand, the thin atmosphere also means less deceleration. On Earth, atmospheric drag slows incoming objects dramatically, sometimes enough that they fall the last portion of their journey at terminal velocity rather than high speed. Mars's thin air doesn't slow things down as effectively, meaning particles can hit the surface at higher velocities than they would on Earth after atmospheric entry size matters enormously here. Tiny particles, dust grains smaller than a millimeter, can survive atmospheric entry more easily than large ones because their high surface area to volume ratio means they radiate heat away efficiently, even as friction heats them. Experiments and models suggest that dust particles in the right size range can survive entry through Mars's atmosphere without completely vaporizing, especially if they enter at angles that aren't straight down, extending their time in the upper atmosphere and allowing for more gradual heating. If we're talking about ice particles rather than rock dust, the calculation changes ice has a lower melting point than rock, which seems like it would make survival harder, but ice particles can undergo ablation. The outer layers vaporize away during atmospheric entry, carrying away heat and protecting the interior, similar to how heat shields on spacecraft work. A sufficiently large ice particle could lose its outer layers to ablation, but retain a core that survives to reach the surface. Now, what about biological material? This is where everything we discussed about extremophiles becomes relevant. For anything biological to survive the journey from 3I Atlas to Mars's surface, it would need to survive several distinct challenges. The radiation exposure in space during the time between being released from the comet and encountering Mars, the heating during atmospheric entry, and the impact with the surface. The radiation challenge is actually not as severe as you might think for this specific scenario, because the time between release and Mars encounter would be very short, hours to days at most. Unlike Panspermia scenarios where microbes spend thousands of years in space, material from 3I Atlas would reach Mars quickly if it reached Mars at all. Radiation damage is cumulative, so shorter exposure time means less total damage. Organisms embedded within dust particles or ice grains would have some shielding, further reducing radiation dose. The heating challenge during atmospheric entry is more severe. Temperatures high enough to vaporize rock would certainly kill any known organism, but we're not necessarily talking about bare organisms exposed directly to heating. We're talking about microbes, potentially embedded within dust grains or ice particles, shielded by the material around them. As the outer layers heat up and ablate away, they carry heat with them, protecting the interior. There's a scenario, not proven but physically plausible, where the interior of a particle remains cool enough for organic materials to survive, even as the exterior is being heated to high temperatures. Laboratory experiments have subjected bacteria to rapid heating events simulating atmospheric entry conditions, and some bacteria survived if the heating was brief enough and if they had some protective matrix around them. These experiments don't perfectly replicate the conditions of Mars atmospheric entry, but they demonstrate that brief intense heating isn't automatically fatal to all biological materials under all circumstances. The impact with the surface is actually potentially less destructive than you might expect, especially if the particle has been slowed significantly by atmospheric passage, and especially if it lands on something softer like sand or dust rather than hard rock. Again, experiments have subjected bacteria to high G impacts and found that some survive. None of this proves that viable organisms from 3I Atlas reached Mars. What it establishes is that the chain of events required for that to happen doesn't violate any known physics or biology, Each step is difficult, but not impossible. The probability of any individual particle carrying viable biological material and surviving the complete journey is undoubtedly very low. But if millions or billions of particles were released into space during the encounter, and if even a tiny fraction encountered Mars, and if an even tinier fraction survived atmospheric entry and impact, you could still end up with viable material reaching the surface. Scientists have attempted to model this quantitatively, calculating what percentage of outgassed material might be captured, what percentage of captured material might survive atmospheric entry, what percentage might contain any biological material to begin with. The numbers are highly uncertain because so many parameters are poorly constrained. Estimates range over many orders of magnitude depending on assumptions. But the central point is that the probability isn't zero, and that's sufficient to make it scientifically interesting. There's one more factor worth considering. Even if no living organisms survived the journey, organic molecules might have. Complex organic compounds are more heat-stable than living cells, and many could survive atmospheric entry conditions that would kill any organism. If 3I Atlas delivered organic compounds to Mars, those compounds could be detectable in Mars's atmosphere or soil, and they could have served as raw materials for any native Martian biochemistry, if Mars has or had its own life. In that sense, material transfer between 3I Atlas and Mars doesn't need to have delivered living organisms to be biologically significant. So the question, could it work, has a nuanced answer. Yes, physical mechanisms exist that could allow material from 3I Atlas to reach Mars, and biological materials could potentially survive under specific circumstances, though the probability of any individual survival event is low. Whether it actually worked, whether the possible became actual during this specific encounter, is something we might be able to determine through direct evidence from Mars. And that's what makes the next part of our story so compelling. In the timeline of this story, the search for answers is unfolding right now, as if you were watching events in that universe play out. The beautiful thing about science is that speculation eventually meets evidence. We don't have to simply wonder whether 3I Atlas delivered material to Mars, whether organic compounds or even biological materials from an interstellar comet reach the red planet's surface. We have the tools, the missions, and the technology to actually look for answers, and that search is underway right now as you watch this Mars, more than any other planet in our solar system besides Earth, has been the subject of intensive robotic exploration. Multiple rovers currently operate on its surface. Orbiters circle the planet constantly, studying its atmosphere and surface. Every one of these missions gathers data that could potentially reveal evidence of material delivered from comets, including interstellar ones like 3I Atlas. Let's start with what we can detect. If cometary material reached Mars, it would leave signatures, chemical fingerprints that distinguish it from native Martian material. Comets have distinctive compositions, different ratios of isotopes than Mars rocks. Different organic compounds than you'd find in Martian soil water from comets has a different ratio of deuterium to normal hydrogen than water found on Mars or in Mars's atmosphere. Organic molecules from comets would have different structures, different complexity, potentially different chirality than any organic chemistry happening on Mars itself. In this imagined timeline, multiple robotic rovers crawl across Mars, each carrying sophisticated chemical analysis instruments. These rovers can scoop up samples of Martian soil and rock, heat them, analyze the gases released, and identify specific molecules. They can measure isotope ratios. They can detect organic compounds at concentrations of parts per billion. If cometary material is present in Martian soil, especially near the surface where recent deposition would be most detectable, these instruments have a chance of finding it. Curiosity has already detected organic molecules on Mars, not proof of life, but proof that complex carbon chemistry exists on the planet. Some of these organics might have been delivered by meteorites or comets over Mars's history. Distinguishing between organic molecules from different sources requires detailed analysis of their structures and isotopes. Analysis that's challenging to do with robotic instruments but not impossible. As analytical techniques improve with each new mission, our ability to identify the origins of organic compounds on Mars improves as well. The atmosphere of Mars provides another avenue for detection. When cometary material enters an atmosphere, some of it remains suspended as fine dust or reacts with atmospheric gases. Orbiters equipped with spectrometers can analyze Mars's atmosphere with incredible sensitivity, detecting trace gases and aerosols. Methane detections on Mars have been one of the most intriguing mysteries of recent years. Methane appears in the atmosphere sporadically, in certain locations, and we don't yet fully understand where it's coming from. Could any of it be related to cometary impacts or material deposition? That's one of many questions atmospheric studies might help answer. There's a particularly exciting development that could provide the most definitive answers: Mars sample return. In this universe, an ambitious sample return program aims to bring pieces of Martian rock and soil back to laboratories, where scientists can analyze them in far greater detail than any rover could. For analysis in sophisticated laboratories, Perseverance rover is currently collecting and caching samples in sealed tubes at various locations on Mars, choosing samples that represent different geological settings and time periods. A future mission will retrieve these tubes, launch them into orbit around Mars, and another spacecraft will capture them and return them to Earth. Why is sample return so important? Because laboratory instruments on Earth are far more sensitive and sophisticated than anything we can send to Mars. On Earth, we can analyze samples with atom-by-atom precision. We can measure isotope ratios with incredible accuracy. We can sequence any DNA or RNA if biological materials are present. We can detect organic compounds at concentrations far lower than any rover instrument can achieve. If cometary material is present in any of the returned samples, Earth laboratories will find it. The timeline for Mars sample return is ambitious. In this imagined era, robotic explorers are already caching samples on Mars, and future spacecraft are being designed to bring those sealed tubes back to waiting laboratories. By the early years of the next decade, if everything proceeds as planned, we could have Martian samples in laboratories here on Earth. Those samples will be studied by researchers around the world, addressing questions about Mars's geology, climate history, and astrobiology. Among those questions will be whether any material from comets, including potentially from interstellar comets, can be identified. There's another approach to finding answers that involves looking at the bigger picture: statistical analysis of Mars's chemistry over time. If comets regularly deliver material to Mars over millions of years, that material would accumulate in the planet's soil and atmosphere. While any individual cometary deposit might be hard to detect, the cumulative effect of many comets over geological time could create measurable changes in Mars's overall chemical composition, particularly in surface layers. Scientists can compare the chemistry of Martian meteorites that formed at different times in Mars's history, looking for variations that might indicate external material deposition. They can study layers in Martian rocks and soil cores, once we have the ability to drill deeper than current missions allow, to see how chemistry has changed over time. The work doesn't stop at Mars. Understanding cometary delivery of material to planets benefits from studying the process elsewhere. We can analyze meteorites that fall to Earth, particularly those known to have come from space without terrestrial contamination, to understand what kinds of organic materials comets deliver. We can study samples from comet missions. The Stardust mission returned samples from a comet's coma, and those samples have been intensively studied for over a decade. Future missions might return samples from cometary surfaces directly. Scientists are also improving theoretical models. Computer simulations of cometary encounters with planets have become more sophisticated, incorporating better physics, better atmospheric models, better understanding of how particles behave during atmospheric entry. These models can predict what signatures we should look for, what chemical fingerprints would indicate cometary delivery versus native Martian chemistry versus meteoritic delivery. As models improve, they guide where to look and what to look for, making the search for evidence more efficient. There's an important principle to understand here. Absence of evidence isn't evidence of absence, especially when we're looking for traces of rare events in a limited number of samples from a few locations on an entire planet. Mars is a world the size of Earth's continents. We've sampled a handful of locations in detail. Even if 3i Atlas did deliver material to Mars, finding that material would be like searching for specific grains of sand on a beach. The fact that we haven't found definitive evidence yet doesn't tell us much. But the inverse is also true. If we do find evidence, if we identify material with the chemical signatures of interstellar cometary origin, that single positive finding would be extraordinarily significant. What would that evidence look like? It might be organic molecules with unusual isotope ratios, inconsistent with Martian formation, but consistent with cometary chemistry. It might be specific mineral grains with crystalline structures that indicate formation in interstellar conditions. It might be noble gas isotopes that match cometary abundances rather than solar system abundances. Each of these would be a clue, and multiple clues together would build a case. The most provocative possibility, of course, would be finding biological materials, preserved cellular structures, DNA or RNA sequences, proteins, that don't match anything on Earth but could plausibly be remnants of biological material from elsewhere. This would be extraordinary evidence, requiring extraordinary validation to rule out contamination or alternative explanations. But it's not impossible. We've found preserved cellular structures in ancient Earth rocks billions of years old. We've extracted and sequenced DNA from samples thousands of years old. If biological materials reached Mars, if they were preserved in the right conditions, if we're fortunate enough to sample a location where they exist, we might find them. The search continues, with each new mission adding data, each new analysis refining our understanding, each new sample bringing the possibility of answers closer. We're living in the era when these questions shift from purely theoretical to empirically testable. The encounter between 3I Atlas and Mars happened at a time when humanity has the capability to actually investigate its consequences. That's remarkable when you think about it. We're the first generation that can seriously search for evidence of panspermia. But let's step back from the technical details of the search and think about the bigger question. What would it actually mean if we found evidence that 3I Atlas, or any comet, delivered biological materials to Mars or any other world? How would that discovery change our understanding of life in the universe? Imagine for a moment that we find definitive evidence. Imagine that analysis of returned Martian samples reveals organic compounds with isotopic signatures that could only come from interstellar space, that match what we know about 3I Atlas's composition, and that contain complex molecules associated with biological processes. Imagine that we can trace those materials to surface deposits dated to the time when 3I Atlas made its close approach. Imagine, in other words, that we prove cometary delivery of biological material actually happened. What changes? Everything. If life, or even just the complex organic building blocks of life, can travel between worlds via comets, then life is no longer confined to its place of origin. A planet where life emerges becomes a seed source for other worlds. The probability of life existing throughout the universe isn't limited by the probability of life arising independently on each world. It's increased by the probability of life spreading from any world where it does arise to others nearby. Think about what this means for our solar system. Earth has harboured life for nearly 4 billion years. During that time, countless asteroids and comets have impacted Earth, and large impacts can eject material into space. Some of that ejected material has reached Mars. We know this because we've found meteorites on Earth that came from Mars, so the reverse journey is certainly possible. If even a tiny fraction of the material ejected from Earth over billions of years carried viable microorganisms, and if even a tiny fraction of those organisms survived the journey, then Mars could have been seeded with Earth life repeatedly throughout history. This possibility flips the question about life on Mars. Instead of asking, did life arise on Mars independently, we might need to ask, is life on Mars related to life on Earth, descendants of microbes that made the journey between our worlds? If we ever find life on Mars, determining whether it's independently originated or related to Earth life will be crucial, and the answer will tell us vastly different things about the universe. If Martian life turns out to be related to Earth life, sharing DNA or similar biochemistry, it suggests that lithopanspermia, transfer via rocks, works, and that our two planets have been exchanging biology for billions of years. Mars and Earth would be part of the same biological system, branches of the same family tree rather than independent experiments in life's origins. If Martian life is biochemically independent from Earth life, using different genetic systems or different amino acids or different biochemical pathways, then we'd know that life arose separately at least twice in our solar system. That would be extraordinary evidence that life emerges readily wherever conditions permit, suggesting that the universe might be teeming with biological complexity. But there's a third possibility that becomes more likely if we confirm that 3I Atlas delivered material to Mars. Life on both Earth and Mars could have originated elsewhere, seeded by comets from beyond our solar system, and we're distant relatives sharing a common ancestor from another star system entirely. This is the most profound version of panspermia. Not just transfer between neighbouring planets, but transfer between star systems, life spreading through the galaxy like seeds dispersed by the wind. This third possibility has deep implications for how we think about life's distribution in the galaxy. If life can travel between star systems via comets, then life might have arisen only rarely, perhaps only once or a few times in our galaxy's history, but spread widely from those few origins, the Milky Way could be biologically connected, not a collection of isolated experiments, but a single vast network of related living systems, all descended ultimately from the same ancestral chemistry that arose somewhere, sometime in our galaxy's past. We don't know which of these scenarios is correct. We might not know for decades or even longer, but the possibility that we could know, that we have missions underway and planned that might actually answer these questions, is remarkable. We're asking some of the deepest questions humans have ever contemplated, and we're doing it with real science, real missions, real evidence rather than pure philosophy. The discovery of life spreading via comets would also fundamentally change how we approach planetary protection. Right now, when we send spacecraft to Mars or other potentially habitable worlds, we sterilize them carefully to avoid contaminating those worlds with Earth life. This is driven by the recognition that we don't want to confuse our search for native life by accidentally introducing our own microbes. But if comets have been delivering material between worlds throughout solar system history, then planets have been contaminating each other naturally for billions of years. This doesn't mean we should be careless. We still want to avoid overwhelming natural processes with concentrated contamination from spacecraft, but it does change the philosophical framework around planetary protection. There are implications for where and how we search for life beyond Mars. Jupiter's moon Europa has an ocean beneath its ice shell. Saturn's moon Enceladus has geysers of water erupting from a subsurface ocean. These worlds are far from the Sun, unlikely to develop life through the same pathways that might have worked on Earth or Mars. But if cometary delivery of biological material is possible, then maybe these distant ocean worlds have been seeded too. Maybe life exists in Europa's ocean, not because it arose there independently, but because comets delivered the seeds long ago, and those seeds found the ocean environment hospitable enough to take hold. The question extends beyond our solar system. We now know that thousands of exoplanets orbit other stars. Some of them are in habitable zones where liquid water could exist on their surfaces. Some of those star systems undoubtedly experience cometary bombardment just as ours does, and interstellar comets travel between star systems carrying material from one to another. If panspermia works, then discovering life on any exoplanet wouldn't just tell us that life exists there. It might tell us that life has spread between systems, that the galaxy is connected biologically in ways we're only beginning to recognize. This is why the encounter between 3I Atlas and Mars matters beyond the specific details of that one event. It's a window into a process that could be fundamental to how life exists and spreads throughout the cosmos. Whether or not that specific comet delivered anything to Mars, the fact that such encounters happen at all, that interstellar objects pass through solar systems and interact with planets, means that the opportunity for material transfer exists. And if it exists as an opportunity, then over the immense timescales of cosmic history, it probably happens as an actuality. We're at the very beginning of understanding these processes. The discovery of interstellar objects like Oumuamua, Borisov, and 3I Atlas is barely a decade old. The techniques to analyze Martian samples with the precision needed to identify cometary materials are still being developed. The missions that will return those samples are still being launched. The complete picture will take decades to assemble, and each piece of evidence will raise new questions even as it answers old ones. But we're asking the right questions, using the right methods, and we're genuinely open to following the evidence wherever it leads. That's how science works at its best. We don't start with the answer we want and look for evidence to support it. We start with the question, gather the best evidence we can, and let that evidence guide our understanding. Whether comets deliver life between worlds or not, the process of finding out will teach us profound truths about biology, planetary science, and our place in the universe. So where does all of this leave us? Let's be completely honest about what we know, what we don't know, and what the realistic path forward looks like. Here's what we assume with confidence inside this story. 3I Atlas, an interstellar comet from beyond the solar system, makes a close approach to Mars. In this imagined universe, that isn't speculation. It's a documented event tracked by multiple telescopes following its trajectory. We know from real physics that comets release material through outgassing as they approach a star, and in our scenario, 3I Atlas is actively outgassing during its Mars encounter. We know that comets contain organic compounds, the carbon-based chemistry that forms the foundation of life as we understand it. We know that some organisms on Earth can survive extreme conditions, including brief exposure to space. We know that material can be transferred between planets in our solar system. We've found Martian meteorites on Earth, proving that rock can make the journey. The underlying physics and biology are established facts in our real world, verified through observation and experiment. In this story, those same principles are applied to the imagined encounter between 3I Atlas and Mars. Here's what we don't know. We don't know if any material from 3I Atlas actually reached Mars. We don't know if any biological materials, living or preserved, were present in that comet to begin with. We don't know if any organic compounds from the comet survived the conditions of space, atmospheric entry, and surface arrival. We don't know if Mars has any life at all, native or delivered, current or past. We don't know if Panspermia has occurred anywhere in the universe, ever. These unknowns aren't failures of science, they're the questions that drive scientific investigation. They're honest acknowledgements of the limits of current knowledge, and they're the motivation for missions, experiments, and observations underway right now. The path forward is clear even if the answers aren't. Mars missions continue to gather data, analysing soil and atmosphere, searching for organic compounds and biosignatures. Sample return missions are in development, progressing through the complex planning and engineering needed to bring Martian material to Earth for detailed laboratory analysis. Comet missions, both past and future, provide comparison data about cometary composition that helps us recognize cometary materials if we find them elsewhere. Each piece of data narrows the range of possibilities. Each analysis either supports or constrains the hypothesis that cometary delivery of biological materials happens and matters. Over time, evidence accumulates, models improve, and our understanding becomes more refined. This is the patient, methodical process of science. Not dramatic revelation, but incremental progress, each small step building on the previous one. What should we expect in the coming years? Continued reports from Mars rovers analysing soil chemistry. Possible detection of new organic compounds or unusual isotope ratios that might indicate external material deposition, improved atmospheric studies from orbiters, and eventually, within the next decade if missions proceed as planned, returned samples in Earth laboratories being studied with unprecedented precision. If we find evidence of cometary material on Mars, it won't be a single dramatic announcement. It will be a series of papers, each analysing different aspects, each building a case through multiple lines of evidence. Scientists will debate the interpretations, propose alternative explanations, conduct follow-up studies. That's how scientific consensus builds, through open investigation, critical analysis, and reproducible evidence. If we don't find evidence, that also tells us something, though it's harder to interpret negative results. Absence of evidence in a limited number of samples from a few locations doesn't prove that delivery never happens. It might mean we haven't looked in the right places yet, or that the processes occur but are too rare to have shown up in our samples. Science often works this way. Positive findings are definitive, but negative findings require careful interpretation. What I want you to take away from all of this is a sense of genuine excitement about the questions combined with realistic expectations about the timeline for answers. We live in an extraordinary time when questions that humans have wondered about for centuries — are we alone? How did life begin? Is life unique to Earth? — are becoming empirically testable through robotic exploration and advanced analysis. But empirical testing takes time. It requires missions that take years to develop and launch, instruments that take decades to perfect, samples that take years to collect and return. The encounter between 3I Atlas and Mars happened recently enough that we're still in the early phase of investigation. We've identified that it's a significant event worth studying. We've established the theoretical framework for what might have occurred. We've determined that investigation is warranted and possible. Now comes the longer, harder work of gathering evidence, testing hypotheses, and seeing where the data leads. This is the reality of cutting-edge science. It's not always fast, it's not always conclusive, but it's always honest. We follow the evidence, we acknowledge uncertainty, we change our minds when new data warrants it, we remain open to being surprised by what we discover. And underlying all of this is a profound sense of wonder at what we're capable of investigating. We can track interstellar objects passing through our solar system. We can send sophisticated robots to other planets to analyze soil and rocks. We can bring samples back from other worlds. We can measure chemical compositions with atom-level precision. We're asking whether life traveled from another star system to Mars, and we have realistic ways of investigating that question. That alone is extraordinary. The universe is vast, and we're learning to read its stories one piece of evidence at a time. Step back for a moment from the specific details of 3I Atlas and Mars, from the physics of atmospheric entry and the chemistry of organic compounds, and consider what this entire line of inquiry represents. Why does humanity devote resources, scientific expertise, and sophisticated technology to questions like whether a comet delivered biological material to Mars? What drives us to ask these questions at all? I think it comes down to something fundamental about human nature. We need to understand our place in the universe. Are we isolated, unique, alone in a cosmos too vast to fully comprehend? Or are we part of something larger, connected to other worlds and other possibilities through mechanisms we're only beginning to recognize? These aren't just scientific questions. They're philosophical questions. Questions of meaning and context that have driven human inquiry since our ancestors first looked up at the night sky and wondered what those lights might be. The investigation of panspermia and cometary delivery of biological materials sits at the intersection of multiple scientific disciplines. It requires astronomy to track comets and understand their origins. It requires planetary science to understand Mars's environment and atmosphere. It requires biology to understand what life needs and how resilient it can be. It requires chemistry to analyze compounds and identify their sources. It requires physics to model trajectories and atmospheric interactions. This convergence of knowledge from different fields is science at its most powerful. Integration of understanding across domains to address questions that no single discipline could answer alone. We're living through the opening chapters of astrobiology, the study of life in the universe. This field barely existed as a formal discipline decades ago, and now it drives some of our most ambitious space missions. The questions it asks aren't settled. We're still figuring out what questions to ask and how to ask them. The encounter between 3I Atlas and Mars will be remembered as one of many natural experiments that helped shape this young science, teaching us what to look for and how to interpret what we find. There will be more interstellar visitors. 3I Atlas is the third we've detected, but as our telescopes improve and our sky surveys become more sensitive, we'll find more. Each one will teach us something about the diversity of objects travelling between star systems, about the chemistry of material formed in different stellar environments, about the frequency of close encounters with planets. Over time, we'll build a statistical understanding of how often these encounters happen, and under what circumstances material transfer is most likely. And it's not just interstellar objects. Our solar system has countless comets in the Oort cloud, the vast sphere of icy bodies surrounding the Sun far beyond Pluto's orbit. Occasionally, gravitational perturbations send these comets falling inward toward the Sun, and some pass close to planets. Every such encounter is another opportunity for material exchange, another natural experiment in panspermia. Over the billions of years of solar system history, the cumulative effect of these encounters might be profound even if individual events are subtle. The bigger picture is that the universe appears to be far more interconnected than we once imagined. Planets aren't isolated laboratories where chemistry happens in isolation. They're part of a dynamic system where material circulates, between planets via impact ejecta, between star systems via interstellar objects, perhaps even between galaxies over cosmic timescales. If life arises anywhere in this connected system, it has pathways to spread, opportunities to colonize new environments, chances to diversify and adapt. This doesn't diminish the mystery of life's origins. Even if panspermia spreads life widely, life still had to arise somewhere, somehow, from non-living chemistry. That original genesis remains one of science's deepest questions. But panspermia adds another layer to the story. It means that understanding life on Earth might require understanding events that happened on other worlds, or in interstellar space, or in stellar systems long gone. Our biological history might extend far beyond our planet's history. The search continues, driven by curiosity, enabled by technology, guided by the scientific method's patient insistence on evidence we don't know where it will lead. We don't know what we'll discover about Mars, about comets, about life's distribution in the universe. But we're looking, and that's what matters. Every time we point a telescope at a comet, every time a rover on Mars analyzes a sample, every time scientists refine models of atmospheric entry or study extremophiles in Earth's most hostile environments, we're adding pieces to an enormous puzzle. The picture that emerges will show us something true about the universe we inhabit, about the processes that shape worlds and life, about the cosmic context of our existence. That's worth the effort. That's worth the decades of patient investigation. That's worth asking the question, what if 3i Atlas delivered life to Mars? Because in asking we learn, and in learning we grow. And in growing, we become worthy of the vast and wondrous universe that surrounds us, that challenges us, and that ultimately welcomes our curiosity as we seek to understand our place within it.